Hello everybody, I'm back. Dr. KK Agarwal, President, Confederation of Medical Association of Asia and Oceania, past National President, Indian Medical Association, and President, Heart Care Foundation of India. And you are watching Medtalks with Dr. KK Agarwal, non-stop 7 p.m. show, 254th edition and 2033 cumulative session. Let's have the headlines of the day. Bangalore sees spurt in under 10 children testing COVID positive. Hong Kong says initial investigation of BioNTech vaccine shows no obvious systemic factors. Maharashtra recorded highest positivity rate of COVID-19. The increasing rate of COVID-19 cases suggests this wave has a more infectious strain. COVID-19 vaccination centers up at inside the Tihar jail. Let's have the topic of the day. भारत में नया म्यूटेशन, क्या वो ज़्यादा डेंजरस है, क्या वो ज़्यादा इन्फेक्शियस है, क्या उसके फिनोटाइप एक्सप्रेशंस हो सकते हैं, इसके बारे में हम जानेंगे इस सेशन में। बट आप सब जानते हैं अतिथि देवो भव, बिना हाथ धो के नहीं किया जा सकता। लेट्स हैव सुमन के एस फॉर सीधा, U फॉर उल्टा, M फॉर मुट्ठी, A फॉर अंगूठा, N for nakhun, or K for kalai. Once we have done that, we will call Dr. Anita Chakravarti. She is a regular visitor to our show. She is a retired professor and HOD microbiology at Maulana Azad Medical College and associated hospitals. She has established the diagnostic virology lab at Maulana Azad Medical College. Her main research interests and contribution have been in clinical virology. She is the recipient of several state and national awards in the field of her work. I present to you Dr. Anita.
Good evening, and thank you, Dr. Agarwal, for the kind introduction. And today I just want to begin by the famous sayings of a Japanese scientist, Dr. Ishii Hiroyuki. And he said, who is the winner, the virus or the man? And he has compared the virus as one of the hackers. And he says the human cell tries to keep the virus out by a password, but the hacker virus uses every method to enter the cell. And humans try to counter the filling of this keyhole. It's either by using monoclonal antibodies or antiviral treatments, therapies, or by vaccines. And nothing could be more apt. Actually, there is an active battle going on between the virus and man. And I'm sure the man would outsmart the viruses. So why am I talking about this? Because this has a close link to today's topic, which is mutations. And I'm going to begin with certain slides. And could I have the first slide, please? Now, this is a well-known picture, how the virus looks. And this is just showing the various structures of the viruses which are present and which are important for all of us to know. And the most important structure here is the spike glycoprotein. And everyone is aware by now that this is the part which binds to the RBD or the receptor binding domain. So this is to the ACE inhibitors which are present in the cell. This goes and binds there, and then the virus goes inside and it replicates and causes infection. The other parts also have their own importance, like the envelope and the nucleocapsid, but the spike protein is the one which has a lot of relevance, especially pertaining to mutations. The next slide, please. Now, this is just showing an open reading frame of the virus. And in the open reading frame of the virus, we can see both the non-structural as well as the structural parts. And the non-structural have all the enzymes like helicase, replicase, papain, proteases, which help during the replication process. And while we have also the structural protein, the spike, the most important. Can I have the next one? Now, there are many types of mutations, and some mutations occur, they are silent, and one doesn't know they are occurring. And these mutations are seen in the viruses. And all viruses have this type of relevant mutations. They happen more in the viruses, which are the RNA viruses, and the ones which are single-stranded. And as we know, most of the RNA viruses are all single-stranded RNA viruses. And they happen in those viruses which have a small, they are smaller in size, the genomic size is small. Now, why does a mutation occur? This is because it's a phase of evolution for the virus, and the virus wants to adapt itself to the new environment, to the new host. That is why the mutation occurs. Otherwise, also, all viruses are replicating. The rate of replication differs, and some mutations will always occur in these viruses, but they are occurring at different levels. For the RNA virus, coronavirus is an RNA virus. It's a single-stranded virus, and it has mutation because of the lack of proofreading ability. The replication mechanism is defective because of this lack of proofreading activity. And it is only not determined, mutation is not only determined by this, it is also determined by host factors and how much pressure the host immune mechanism is giving, how much selection pressure the viruses are undergoing, just like bacteria, like they undergo selection pressure when too much of an antibiotic is given and they develop resistance. Similarly, here also the selection pressure comes into play. Variation can also occur at the human host level, and mutations are seen in different regions of the viruses. But the most important, which is seen and which is now the talk, is the spike region. But there would be, you never know, there could be mutations coming up in the other regions. What is important for us is that if the mutation occurs, does it have any effect on the host? If they are silent mutation, no, we are not bothered. There could be missense, there could be nonsense, that is not bothering us. But what is bothering us is frame shift mutation, in which there is deletion or there is insertion of some amino acid sequences. So mutation is at the level of the amino acid sequence, and where the virus, these amino acids making the protein. Mutation is at the spike level, usually the S1 element, because the S2 is the fusion element. So the S1 element shows the different types of mutations. And here you can have the amino acid deletion or insertion, which could lead to the spike protein having a different type of arrangement of the genome. And if that happens, then it is indeed very dangerous, because then it would have an advantage over the host cell. Could I have the next slide, please? So now, when we talk about mutation, actually what it refers to is a virus genetic sequence. There will be some change in the genetic sequence, as I've already told. And when such a mutation occurs, and this results in some change in relation to infectivity, in relation to virulence, then we call it as there is a variant of the original virus. And this variant is a variant of concern. That's why it's written as VOC. And the mutations, they result in the virus. And what happens is either it's more transmissible, it is more virulent, more infectious. It could have effect on vaccines. The efficacy of the vaccine could be lower with the variation, and therapeutics may not work to that extent. So when these variants are there, they increase the risk to humans, then they are called as VOCs, or variant of concern. And there are many infections can occur, multiple infections in a population. And when such a multiple infection occurs, then there is a more reason for having virus mutating, and this mutation rate increases. The next slide, please. Now, this is just the examples which we have been reading, which the early mutation almost started, the detection started early in 2020s, and we are still detecting double mutants now in 2021. So these mutations occur, and they are called as N501Y mutation. There are still others, N440k mutations, E484k mutations. There are double mutations like E484Q, L4452R. So what are these mutations? What do they have effect, and where do they occur? Can I have the next slide? So, as can be seen, there's a big open reading frame with a 5 UTR region and a 3 UTR region. And here, the arrow is focused on the spike because most of the mutations have been seen in this region. The other mutations which occur, newer information is coming about them. But presently, I'm going to talk about the spike mutations. And amongst the spike mutations, what is seen is that the domain which is mainly affected are the receptor binding domains. And this receptor binding domain, the amino acids in these domains are affected. And when they get affected, then that mutant like 501 would be very near the RBD. It would be in fact a part of RBD. Similarly, the others are also part of RBDs, but there are some mutations in which along with the mutation there is deletion. So when this deletion occurs, then it becomes even more dangerous because part of the amino acid is not there. The defective protein which is formed could have more invasive or infectious effects. Can I have the next slide, please? So when mutation occurs, what happens? This is a very simple slide. Things are much more complicated. So there is a variation in the amino acid. So either a mutation could lead to asparagine to tyrosine mutation, like in the case of 501Y, because this is the one which is most common, I'm repeatedly telling. Or it could be glutamate to glutamine, E484Q. Still other mutations, you can have the replacement of the amino acids. The replacement of the amino acid leads to a new structure of protein which is being formed. Can I see the next one, please? Now, we started first with the UK, then we had the South Africa, then we had the mutants from Brazil, then we still had other mutants coming into play, and those were the ones which were seen in Denmark. Then there were mutations which occurred in USA, and then of course we have mutations occurring in India itself. So the first one which occurred was the B117 or the 501YV1. V1 is a variant number 1. When it changes, it becomes, to make it more unambiguous, we call it V2 or we call it variation 2, variation 3. Now B501, this 501YV1, this had some deletion in it, and it was seen that this mutation was more infectious, was more lethal, and this occurred for quite some time in UK. After that, there was another mutation that was seen in June. It was seen in USA, and that was the B1429. This was the lineage, and this lineage, it was seen that it reduced the neutralizing antibody. That means it would have an indirect impact on the person recovering soon, as well as on the efficacy of the vaccine. Then in September, there was a mutant which occurred that was seen in Denmark. South Africa, 501 V2 mutant was seen, and also E484K mutant was seen. The same mutants were also seen in Brazil as well as in Japan for some time, and there was another mutant in Japan which was a virulent mutant, but they could contain it. And in UK, and what was seen was E484 recently in December 2020. So these are the major mutants which have been seen. There are thousands of mutations going, but these are the mutants which have caused various effects of increased spread of infection, increased infectivity, neutralizing antibodies not being generated, so the prolongation of the disease and such type of symptoms. What we are interested is, if the mutants are there, what type of manifestation, what type of symptoms they would give, if they would give more exaggerated symptoms. So these were the ones which could be seen. The next one, please. Now, N501Y mutation, I would like to elaborate a little bit about this. This was seen first in UK, and this also appeared in Brazil called as P1, and then in South Africa, a variant appeared, and it was called as B1351. Now, this allowed the virus to bind to the ACE receptors. So once it allows the virus to bind to the ACE receptor, then automatically it can get more access into the body, it can multiply, it becomes more transmissible. This was responsible for reinfection after vaccination. And I remember that in the initial stages, when we were asked about reinfection, it was also told that reinfection is anecdotal. But now we see that reinfection is occurring. So this virus is coming with so many different things. Where we think it will not happen, it is still occurring. So here, this was responsible for reinfection after vaccination. And it was also seen in India, in some cases, in Punjab as well as in Delhi. The next one, please. N440k mutation. This was said to be a little bit more milder, and it led to the body escaping the neutralizing antibodies, which are very important for us when we develop the immune response. It neutralizes the virus. So even if the antibodies were there, they could not. So this is called as a escape mutant. This was found in nearly 34% of the 272 SARS coronavirus 2 genomes, which were analyzed from Andhra Pradesh. I'll be talking about our genomics later on, about the genomic centers which are working here. And the variant also was seen in Karnataka, Maharashtra, Telangana. These states are close to each other, so the spread was seen because of people moving from one state to the other state. The next slide, please. Then we had the 484k mutant. This was found in South Africa and called as Brazilian P1, and the other one called as B1128 variant. This could efficiently reinfect people who were infected with the original virus, and it was seen to weaken the immune response. The publication which came out in PMID that related to all this, and the impact, the longevity of the neutralizing antibody was much lesser. This was what was analyzed and seen. The next one, please. So each of the mutants had its particular feature. Now this is about the double mutant we are talking about. The good part is that we have to know much more. So let's see what will be revealed, because much more studies have to be done, much more data has to be generated to actually say what it is causing. So some 10,787 samples from 18 Indian states, they showed with these 771 cases of these variants. And double mutation in the virus spike protein was seen. And it was contemplated, it was thought that most probably this double mutation would lead to escape mutants. And what do we mean by escape mutants? They would escape from the immune mechanism of the body and they would cause infection. However, though they are more transmissible, because now we see the second spike coming and the transmissibility high. However, we need much more data and many more samples are being analyzed. And there are more double mutants also have come into the picture. So you don't have one type of virus, you have more types of viruses in the body, and they could cause more severe type of infection. But this data has to be analyzed to exactly find what is the effect. Whether the prior infection, the primary infection with the original virus gives some type of cross-protecting antibodies, because many times in a virus, it is seen that you develop cross-protecting antibodies. But sometimes it is also seen that the cross-protecting antibodies can cause antibody-related cell-mediated immunity, what is called as the ADCC, cell-mediated cytotoxicity. So it has to be seen what exactly happens. So these cases are being followed up. The next slide, please. Now, there is a group which has been established, and they are actively working on genomics. And this is called as the INSACOG, Indian SARS Coronavirus 2 Consortium on Genomics. It's a group of established national laboratories, like in India, in Delhi we have ICGEB, NCDC. These are already working on genomics. They have a good genomics setup. NII, Trans-THSTI, these are the labs, NIV Pune. So these are some of the labs. There are others in the south which are also in this INSA-COG group, and they are doing the genomics, and they have been established by the Ministry of Health and Family Welfare, Government of India. And this was started seeing the amount of variants which are occurring. And therefore, it was realized that when they analyzed these 10,787 positive samples, that there were 771 variants of concern. And these variants and a new double mutant variant have till now not been detected in numbers sufficient to exactly establish the direct relationship or explain the rapid increase in cases in some states. However, they are being looked at upon actively. They are being followed up actively to see what is the result. And as we notice, whatever cases are occurring, infectiousness is high, transmissibility is high, but the lethality is lower. So that is another thing which is going to be studied upon. What is the reason of that? Are these mutants less virulent? Has the body developed some type of immune response? If so, which is the antibody response? Which are the cells coming into play primarily? Has the CMI come into active play? Because ultimately it's going to be the CMI, the T helper cells coming into play. We'll even have the immunoglobulins. So these are the other factors which are being studied. And there are a lot of reports on that, which I will not be covering in this. Now, the next one, please. So what was the necessity of doing genomic surveillance? Was it really essential that we should have done genomic surveillance? Well, I think it was done in the nick of time. And in fact, if it was done earlier, it would have been better. Why? Because we have people coming from international places. And it has been found that they come with these infections. And at present, what is being done is that when you get these type of strains, they are being isolated. Their contacts are being traced. They are being tested. They are being treated. And they are also isolated, so that we can, restrict the infection, contain the infection, which is very imperative. It should not be that suddenly you get a highly infectious, highly transmissible, highly lethal variant and may not be that effective against the vaccines. So we will have the problem which will be highly magnified. Therefore, at present, this is the way of tackling these cases. So this started, and it helps in the ability to contain the infection, also to see whether it causes milder or severe disease in the people. The immunological parameters are being worked on these cases to see if they are evading specific diagnostics, because there is a lot of chance of evading the diagnostic test. Not all tests test all the required genomes. There could be some tests which only test spike. Now, if there is a mutation in spike, maybe you will not detect it. There are some which only test envelope. So it's important that if such a thing occurs, that maybe multiple genes would be tested. The problem comes, it becomes very expensive. It may not be economic, but then one would know precisely what gene to test. Even if a mutation impacts one of the targets, other RT-PCR targets will still work. However, there are some tests which are there which only have one target, so they don't pick up. Can I have the next slide, please? Now, these mutants, they could have, or variants, they could have decreased susceptibility to monoclonal antibodies, because that's one mode of therapy. They could have decreased susceptibility to the antiviral which are being used, remdesivir and flavipiravir. All these are the ones which are being used. They are not the specific ones, but they are showing some results. The mutants could also evade natural or vaccine-induced immunity, because both natural infection with and vaccination against SARS coronavirus produces a polyclonal antibody response. So that would target the spike, but hopefully it would also target the other proteins. The virus could accumulate significant mutation in the spike protein to evade immunity induced by vaccines by natural infection, which would be a dangerous thing. So among these possibility, the ability to evade vaccine-induced immunity is the one which we are most concerned about, because we have good vaccines. We are vaccinating a lot of people, and lots of newer vaccines are going to come into play. Single-use vaccines are going to come into play, and vaccines using different vectors, vaccines using different techniques, these are also going to come into the market. So definitely there is no definite evidence that this evasion of vaccine-induced immunity is there, but scientists are concerned, people are concerned, and therefore a closed evaluation is very much needed. Can I have the next slide? So with these words, I would like to end that a lot is being done, a lot needs to be done, and we are combating an extremely smart virus. As such, viruses are very smart, like the influenza virus. We know that H1N1 virus, vaccine against H1N1 virus, will not neutralize H3N1. So we have vaccine in our hand. So if such a phenomenon even occurs, we can rapidly change the structure of the vaccine or tweak some elements in the vaccine so that we have the vaccine against the disease. But up till date, most of the vaccines, they have efficacy. They may lose some efficacy against some variants, but still they are efficacious. So with these words, I would like to end this particular talk on mutants. I have actually given a very brief talk and not gone into very much details about the mutants.
Thank you, Anita ji. I will now take it up into your slide which we have shown and discuss that slide again. आपने ये स्लाइड दिखाई थी. इस स्लाइड के अंदर में मैं अपनी कुछ लेटरल थिंकिंग से ऑब्जर्वेशन देने की कोशिश कर रहा हूँ. नीचे हम देखते हैं प्रोटीन को S1 और S2 को. तो मेरा एक लेटरल थिंकिंग है कि म्यूटेशन लेफ्ट की ओर जाती है हेड की तरफ. SP is the head, CP is the tail, 5 UTR and 3 UTR. तो जो म्यूटेशंस हो रही हैं वो सारी लेफ्ट की तरफ हो रही हैं खतरनाक होने के लिए. लेट अस सी. सबसे पहले 681H वाली आई जो 679 के बगल में थी, ये यूके वाली स्ट्रेन में है. उसके बाद में आया 614 जो प्रेजेंट वाइल्ड स्ट्रेन है, वो लेफ्ट में गया इसके. फिर 501 आया जो आरबीएम के बाद वाले आरबीडी में है. 501 वो फिर फर्दर लेफ्ट हुई. जैसे जैसे ये लेफ्ट की तरफ जा रही है वैसे वैसे नई स्ट्रेन आ रही है और हर नई स्ट्रेन फर्दर लेफ्ट की तरफ है. फिर हम देखेंगे तो दो आए, दोनों आरबीएम में आए, 484 और 484 आया. सो 484 के बाद में 484 आया, फिर 452 इंडिया में आया, फिर 417 साउथ अफ्रीका और ब्राजील में आया. सो देयरफॉर इंडिया का जो आया 452, it is between 417 and 484. Therefore, it has to be less dangerous than the South Africa and Brazil. Then 6970 and 144 deletion आया, फिर 6970 deletion आया. तो 144 से deletion से ज़्यादा 6970 खतरनाक हो गया. सो इसको देख के एक प्रेडिक्शन सामने नजर आता है, अगर हम इसको देखें कि जितने भी वेरिएंट ऑफ कंसर्न हो रहे हैं उनका जो म्यूटेशन है वो लेफ्ट की ओर जा रहा है। जितना वो लेफ्ट की ओर जाएंगे उतना वो म्यूटेशन घातक होगा। एनटीडी के अंदर में R1, R2, R3, R4 हैं। उसके अंदर में जैसे जैसे म्यूटेशन R1 की तरफ जाता जाएगा और जैसे जैसे म्यूटेशन उसका लेफ्ट की तरफ जाता जाएगा वो खतरनाक होता जाएगा। इसका मतलब है जो हेड पोर्शन है और टेल पोर्शन है वायरस का, उस तरफ में जो स्पाइक प्रोटीन है ऊपर की तरफ और नीचे की तरफ वाला जो S2 है वो मेम्ब्रेन को पकड़ने के लिए है, फ्यूजन के लिए, इसलिए फ्यूजन पेप्टाइड्स हैं उसके अंदर में, ट्रांस मेम्ब्रेन डोमेन है उसके अंदर में। उसके जो म्यूटेशंस है वो ज़्यादा मैटर नहीं करते। केरला में जो म्यूटेशन आया था वो या शुरू मतलब वो जो 1-2 समथिंग था। सो मेरा ये मानना है कि भारत का जो वैक्सीन है 452, क्योंकि ये 417 और 484 के बीच में है, तो ये साउथ अफ्रीका इस वक्त में जो ब्राजील में मोर्टेलिटी हुई, साउथ अफ्रीका में मोर्टेलिटी हुई, शायद भारत में उतनी मोर्टेलिटी ना हो। ये एक मेरा ऑब्जर्वेशन है। दूसरा मेरा ऑब्जर्वेशन है कि भारत के अंदर में ग्लूटामीन आया क्यू के अंदर में, भारत में ग्लूटामीन आया क्यू के अंदर में, और L52 के अंदर में आर्जिनिन आया, और उनके यहाँ पे 452 एन एस्पार्जिन और एन थ्रियोनिन है जो उनके अंदर में आए हैं। एस्पार्जिन एंड थ्रियोनिन मे बी दे आर रिस्पॉन्सिबल, अमाइनो एसिड मे बी रिस्पॉन्सिबल फॉर मोर म्यूटेशंस। सो मेरा ये एनेक्टोडल ऑब्जर्वेशन है कि जैसे जैसे म्यूटेशंस लेफ्ट की ओर जा रहे हैं ज़्यादा खतरनाक हो रहे हैं। सो दैट कैन बी प्रेडिक्टिव। आई डोंट नो वेदर इट इज़ ए वायरोलॉजी के अंदर में नोन फिनोमिना है कि नहीं है। दूसरा जो मैं बता रहा हूँ, मैंने वो स्लाइड ढूंढ ली कि भारत के अंदर में इन्होंने इन एरियाज ऑफ कर्नाटका, महाराष्ट्र, मध्य प्रदेश, न्यू दिल्ली एंड ओडिशा में इन्होंने 2086 केसेस के अंदर में इन्होंने 34 जीनोम पाए जिसके अंदर में ई जीन के अंदर में डिफेक्ट था। तो ई जीन म्यूटेशन वाज सीन इन 34। आउट ऑफ दीज 34, 8 केसेस के अंदर में ई जीन टारगेट फेलियर था, और उन्होंने ये डिसाइड किया इट वर्क्स आउट टू बी 32,500 जीनोम्स with large deletions in 8.4 million cases detected across the country। यानी कि 32,500 केसेस मिस हुए होंगे भारत के अंदर में। ये जो स्टडी है ये बी कृष्ण कुमार, पृथ्वी सागर एंड परवीन राय के इंद्रानी एंड दिस इज मद्रास मेडिकल मिशन से ये स्टडी उस वक्त में आई थी जिसके बारे में मैं बात कर रहा था। तो ये जो फिनोटाइप है जिसमें ई जीन टारगेट फेलियर हो रहा है, this has to be different than the present genomic which is in S protein। और एक तीसरा फिनोटाइप है जो अभी बॉम्बे में दिखाई दे रहा है, अभी जो बच्चों में दिखाई दे रहा है जैसे आज आया है कि 10 बच्चे में ये केस आया। कौन सा जीनोम है मुझे नहीं पता, इसमें आप कुछ बोलना चाहेंगे?
No, in the children, E gene failure was detected, yes, as per the article, and that would have impact when you're testing. So one would miss a lot of E genes. So unless you have the right kits which have multiple genes, the virus will not be detected. The presence of the virus genome will not be detected. Now, regarding E gene, spike gene is the most important for pathogenicity. And the E gene comes on the virus when it is budding out of the cell, because usually the envelopes are part of the cells. So yes, it would be good to study how much virulence is there, but as such, E gene would not cause so much of a virulence. In fact, E gene causing having the virus without an envelope, that would be a sort of a defective virus, you know. So that's what I think.
This observation कि left की तरफ ज़्यादा serious हो रहा है.
See, virologically that definite why it happens.
No, क्या ये ऐसा कोई known phenomena है या मेरी observation है?
No, that is not a very known phenomenon. That is more of an observation. But yes, it would be good to see in future if such a thing is occurring. It would be good to see. But yes, I am also worried about those children and in Bangalore, and I'm waiting to see which are the strains which are causing infection in these children, and how infectious are they, manifesting infections or not. That is what is a slightly bothering. Or whether these
पिछले एपिसोड में बच्चों में तो हमने बहुत केसेस देखे थे. I don't think so कि बच्चों में होना है. I have seen बहुत बच्चों में मैंने दिल्ली में पेशेंट्स को देखा था.
With clinical manifestations.
नहीं, हमारे यहाँ पे तो total manifestation. I have seen all symptoms I have seen in children, but children के अंदर में मेरी observation थी the symptoms last only for 2 to 3 days. They have loss of smell, loss of taste, fever, cough, cold, सब देखे हैं मैंने बच्चों में. दिल्ली के अंदर में मैंने बहुत बच्चों को देखा है. लेकिन अगर कोई स्पॉट आ रहा है, सिर्फ बच्चों में आ रहा है, बड़ों में नहीं आ रहा, तो स्पॉट कहलाएगा. बट व्हाट वी सॉ देयर विद द होल फैमिली, बच्चे भी इसमें इन्वॉल्व थे, सबको हो रहा है.
It was initially thought कि children are asymptomatic, but they are causing.
I saw lots of symptoms in the children.
So they are causing children.
कल ही शाम के मेरे शो में 5 साल की बच्ची को उसके पेरेंट्स ने बताया लॉस ऑफ स्मेल, लॉस ऑफ टेस्ट हुआ था पूरा.
So if this is occurring, then that's another reason why people are trying to see whether they can do studies on vaccines in children. So actually these studies have been put forward. So they would be doing studies on children to see whether the next age group is going to be below 18, and then later on in the children, and if they can vaccinate the children. The whole idea is to vaccinate all the population because of such cases. And fortunately, we are not seeing too much of Kwasaki in India. That is extremely fortunate.
जाते जाते कुछ क्वेश्चन ले लें. रजनीत कुमार राजपूत, why is mortality less in India than other countries? This is what we had postulated in the session. मेरी वाइफ का गॉल ब्लैडर केस. वीरेंद्र शर्मा जी, आप 8 बजे वाले शो में आ जाइए. I will take you in. जब तक आप हमारे साथ हैं, हमें म्यूटेशन से क्या फर्क पड़ता है. ये मनोज जी बोल रहे हैं. भवनीत सिंह जी, मैं स्टेरॉइड रेजिस्टेंट पोस्ट डेंगी ITP 2 years back, treated with rituximab. Now platelets are in low normal range. Is it safe to get vaccine? Vaccine से definitely याद रखिए, you have to calculate your risk versus benefit. There is a theoretical risk your ITP may get precipitated. So you talk to your doctor. If you are taking vaccine and if you develop ITP, then immunoglobulin should be ready. My wife has gallbladder stone. Can she take vaccine? Yes. Good evening, sir. My father's CRP is 2. My mother's CRP is 1.25. Colchicine लीजिए और वैक्सीन लीजिए. रीइंफेक्शन का तक कितने केसेस इंडिया में हुए हैं? बहुत कम है. That the coronavirus will be mutated according to environment. Yes. राजपूत जी, बट आपकी थोड़ा डिटेल में बताइए. Our research team had already announced that the coronavirus will be mutated. It has to mutate. You can't stop it. I was trying to contact you to take your suggestion. Hypertension, BP, sugar. BP आपका अनकंट्रोल्ड है. स्टैटिन, एस्पिरिन, कॉल्चिसिन है, बट शुगर की दवाई को थोड़ा सा कंट्रोल करो, फिर वैक्सीन लो. Can't like influenza vaccine is multivariate. The COVID-19 multivariate killed virus can be procured. I agree with Arun Garg ji. Ultimately वही होगा. Universal vaccine बनेगा कोविड का, like we have Pneumo 23, Pneumo 13. मल्टीपल स्ट्रेन्स का किल्ड वायरस अल्टीमेटली बनेगा. हैप्पी होली टू एवरीबॉडी. और भी सवाल हैं, बट बाकी सवाल लोगों से मैं रिक्वेस्ट करूंगा कि आप 8 बजे वाले शो में आ जाइए. आज हमारा शो 8 बजे भी है और 9 बजे भी है. आप उस शो में आ जाइएगा. सो लेट मी थैंक डॉक्टर अनीता टू बी हियर. आई ऑलवेज लर्न अ लॉट ऑफ थिंग्स फ्रॉम हर. एक सर्टिफिकेट आप दिखाएंगे मनमीत जी, सर्टिफिकेट ऑफ एप्रिसिएशन. ये मैडम अनीता को आप लोगों की तरफ से हम ये भेज रहे हैं एज ए डिस्टिंग्विश्ड स्पीकर. एंड दैट्स ऑल फॉर टुडे. इसको हटा लिया जाए. दैट्स ऑल फॉर टुडे. विल बी बैक टुमारो 7 PM विद अनदर शो Keep watching Medtalks with Dr. KK Agarwal. Namaskar. Namaskar, madam.
Thank you. Happy Holi. No playing. Play safe at home. That's my last message.